All right. Well, I am, thank you all for joining today um, for the VICC call. I'm Beth Pitel, and I'm here with Chen Wang, and we're the co-leads for the VICC and AMIA Complex Variant Working Group. And we've been making some progress lately, so we thought it would be opportune for us to come and give an update on what's been happening in our working group in the last year or so. And we've been very busy working toward a manuscript, but we'll get into that too. So for today, we're going to talk about the different goals of the working group, talk about the manuscript we've been writing, kind of the different angles that we've been looking into for the manuscript, explaining the challenges in annotating complex variants and discussing them. And then also talk about briefly next steps for phase 2 of the working group. And so I tried to illustrate kind of our challenge here, where sometimes if we're talking about a simple variant, we can use the same language to talk to a clinician or to explain what this is to a database stakeholder. And so we don't have to have different ontologies between the two. But as the complexity of the variant increases, the difference between these two ontologies often is greater. So for instance, where a clinician would get more out of seeing a TP53 deletion, we might need to explain that in genomic coordinates for like a database stakeholder or even like an assay dependent nomenclature. And then even increasing in complexity, in our practice, we saw a very complex rearrangement involving an ETV6 PDGFR beta fusion, where only the ETV6 PDGFR beta fusion is important to a clinician. However, if we're going to talk about a full characterization of that complex variant, it gets so lengthy and not really human readable. So this is the challenge that we face, or one of the challenges that we face when looking at complex variants, which we preliminarily defined as structural variants and copy number variants. So our working group has 34 members who have kind of been in and out of the team. These members are representing clinical laboratories, electronic health records, commercial laboratories, and academic institutions. We have 4 continents represented, and we're currently, as I mentioned, writing a manuscript. And so the goals of this working group first were to define what a complex variant is and what our first investigation would be in looking into the challenges or setting the landscape and describing the landscape around complex variants and the challenges that we see in cataloging complex variants and communicating complex variants across assays, and then also across different ontologies. So just explaining what the challenges are in annotating these. So as I mentioned preliminarily, we've defined a complex variant as copy number variants and structural variants, with the understanding that there may be single events that compound complex these different complex variants and create a molecular profile. So the group is trying to study and understand the current landscape and the challenges from 3 different vantage points, one being bioinformatic tools and artificial intelligence, one being nomenclature for different professional roles and different assays, and then also knowledge-based infrastructure. So how do we communicate and search previous experience? So Dr. Chen Wang, who's on the call today, has kind of been leading the bioinformatics vein of this, looking at different bioinformatics pipelines and AI. So some of the challenges in bioinformatics arrive from short read sequencing alignment. There tends to be a balance that needs to be struck between the sensitivity of an assay and the computational burden. And so depending on the different callers or aligners that we use, that balance may shift one way or the other. Additionally, there's complexity in the reference genome, usually in the form of repeats, and this can cause alignment issues. As you know, we only have A's, G's, C's, and T's in the genome, so we can have things like microhomology, or we can have macrohomology with large repeats that happen throughout the genome. Another challenge is variant filtering and knowing when to subtract noise or known polymorphic CNVs and SVs. And there's been quite a bit of traction for solutions or aids in helping out with these bioinformatic pipeline, with these challenges that we see in bioinformatics, including having a pangenome reference or repeat aware structural variant callers like in Surveyor or Survey and Dell. Long read sequencing really helps to define some of these unique sequences that maybe sit in the middle of otherwise repetitive regions. So as long read sequencing becomes more readily available, that can help solve some of these issues. Publicly available databases and annotations can help try to assess these issues, and then the AI and deep learning algorithms can detect complex variants and different patterns as well. So we had discussed this as a group. Chen, do you have anything else to add on this slide?
Bioinformatics. So even with jet lag, I can amplify that further. For the bioinformatic part, I think our write-up central around three aspects. One is variant calling, complex variant calling, and how much that influenced by the issue or limitation, as Beth explained. Because, like, for example, CNV, I think it tend to happen in the repeat or like homology region. So alignment become critical, and understand the limitation reference become critical. We have one of the leading bioinformatician from Hong Kong group. They developed several, remedy approach to dealing with the limitation. That was very instrumental. So first aspect is variant calling, the challenge and the current some kind of resolution for dealing with short read sequencing-based complex variant calling. The second aspect is annotation. I think Jack is in the call, and he did a fantastic job, because the annotation of complex variant range from technical aspect to bioinformatic, and then to the biological aspect, like which gene is disrupted, what kind of disruption, to the clinical annotation. In order to convey the annotation message across like a different group of audience, that's a non-trivial topic. Probably potentially that could also influence knowledge representation and knowledge base. And third aspect, which is AI, it's not just trying to catch up the buzzwords. AI has been relatively successful for encoding the nucleus or single nucleotide level like SMP or SNV. There are high-profile papers to publish on that. However, to generalize the comparative AI work to the complex variant, we require like a different consideration, not only about the reliability of data, but also lacking of the nomenclature we can sufficiently, standardize available complex variant result. AI indeed, like we at the very early stage for complex variant comparing to the simple variant or base pair level of the AI encoding. So bioinformatic part, I think similarly to other parts, by itself, it could be a paper, we were trying to squeeze or like condense them to a comparable section and speak for the interaction with the nomenclature need as well as, to the knowledge base.
Mm-hmm. Thank you. Yeah, thanks. And then I'll just add a couple details in the next couple slides, just showing some figures that some of the members have put together. So Dr. Kim Sung kind of had this really helpful illustration. So imagine you have a deletion that encompasses part of a couple repeats here. And so the deletion takes about half of this repeat and half of this repeat. However, the read pairs in real life are going to say that this looks correct, because if this sequence on this side of the breakpoint and this sequence on this side of the breakpoint still match the reference, then those reads are going to appear to be concordant with the reference genome. Even though normally we would expect those reads to map to their respective locations, but repeats are tricky. And so the alignment on the reference genome appears to be aligned correctly. So a couple ways we could resolve this. So sequencing coverage in the data can help, but we can't discern which repeat is involved unless there is unique material embedded in the sequence. And if there is unique material between the repeats, that may be detected as a loss as well. Also, if we have contiguous repeats like this and the deletion, we have the same kind of problem where we're going to have a similar junction here as we would expect in a normal reference genome. So if reference genome has 3 repeats and one of those repeats is deleted, your junction or your spanned reads at that breakpoint of that deletion are going to look just like they would at the end of that 3rd repeat. And so sequencing coverage may help if we're deep enough, but we can't discern which repeat is involved unless there is unique material embedded and sequenced. So it's kind of a unique problem here that we have with repeats. So some bioinformatics challenges. So pipelines and tools need to be easy to install and use common language that can be widely used and embedded into other softwares. We need these pipelines and tools to be storage efficient and fast, as we're usually working with very high throughput technologies that store lots and lots of data. It helps if they're open source, and we need them to be interoperable. And so groups like GA4GH Burst, Variant Representation Specification, have been working on different data models that can help do this. And also, some of these challenges may be alleviated by using AI techniques. And what we learned was that image recognition is a little bit more advanced than other types of AI. So if we push our data into more of the image type model, then the AI can better digest that data and help us out. And then I wanted to acknowledge Jag. He built a tool that is on a GitHub repository that can help track different features of publicly available and commercial tools. And we're going to add this as a figure to our manuscript that we plan to submit to the Journal of AMIA or JAMIA. And these can be kind of like tracked in real time and changed as needed. So I don't know, Jag, do you have anything to add to this? I know I see you on mine.
It's too colorful. Jack, say something.
Oh, you're still kind of muted. I'm not hearing you, Jag, but this is this is really a nice tool where it talks about the different kind of needs and that are being met. So it breaks down informatics needs, genomic context, different guidelines that are used if we talk about pathogenicity and treatment guidance, and then along all of these different tools. So it's pretty neat.
Can you hear me now, Beth?
Oh, I can. Hi,
Okay,
Jag.
awesome. Sorry. Yeah, so one thing I just wanted to add was like we could benchmark a few tools here and represent the features, but there are many more tools that we couldn't. So we do acknowledge that there might be much more in the field, but we picked the popular ones as of now.
Right.
Yeah,
Yeah. So this
If
is
I
not an
can, if I
exhaustive
can
list.
add like from top
Yep.
to the bottom, like informatics need, that's more like, computer or bioinformaticians like convenience, right? And then in the
Hmm.
middle, the genomic context, that's really related to the biology and nomenclature. How do we represent or annotate each complex variant sufficiently to understand their impact, including the bias? For example, it may overlap with segment duplication. Then towards the bottom part, we begin to get into the clinical significance or relevance. So you can say that across the column, we have so many tools to be developed. I mean, not to mention, probably we have more tools will be developed without appreciate the complexity of the annotation across, like a bioinformatic, a technical aspect to the biology to clinical side. It's very difficult to benchmark the new tools or evaluate existing tool. I guess that's the unique contribution, as Jack's graph, and he made this graph interactive and updatable, has a GitHub repo associated with that. I think that was Howard's suggestion, right?
Right.
I think it's a critical contribution.
That's great. And it's nice that it's interactive because these tools are improving all of the time. So being able to update and kind of show what features are available at the present time is really nice.
And plus, like you think about, in the middle, the database, and the database themselves will get updated through the time.
Right. Yeah. And some of these databases or knowledge bases almost could fit as a column too. But yeah, we won't get too far into the weeds with that. But this is a really nice resource that we have and that
Yeah,
Jag had built.
maybe that's like a perfect place for me to stress again. That's the reason the bioinformatic, even the bioinformatic orientation alone will have interaction with nomenclature, that how we represent, and versus the knowledge base. Because sometimes annotation, bioinformatic annotation tool need to grab the information from the latest knowledge database to annotate accordingly.
Hmm.
So it's not siloed by itself.
Great. Thank you. So next I'm going to talk a little bit about nomenclature. And you may have seen some of these slides previously, but I think it's kind of nice to lay out that nomenclature can vary by assay. And so if we're talking about a BCR-ABL fusion, which is likely going to be on reports as that because that's what a clinician wants to see with like a CML diagnosis or a BALL diagnosis along with like an isoform. But by chromosomes, a different nomenclature is used. ISCN is used. And so if we have conventional chromosome analysis, it might look like this. Microarray analysis may see an unbalanced rearrangement if we have the additional derivative chromosome. If we have a balanced rearrangement, a lot of times microarray isn't going to be able to see that. That's going to be cryptic, but we would be able to see something by chromosomes. And then I created this. So the biological context is BCR-ABL fusion, but there's a lot of, this is just another way to look at this. we could see a lot of different iterations of this depending on the assay and a lot of different ontologies explaining the same thing. So by chromosomes, we're looking at like a T922. By fish, we might use this nomenclature. By microarray, again, if it's balanced, we may not see it. So it may be a normal microarray. By NGS, we'd see a BCR-able fusion. VICC fusion specification, which is endorsed by HGMD, might look something like this with an intercalating sequence or a linker sequence. And the fusion data model would have a list of multiple specifications needed to catalog this fusion in databases. So I didn't list them all out here, but just imagine kind of filling out a form with lots of different attributes that would go along with this fusion. So lots of different ways to talk about the exact same thing. And then GA,
I thought
yep.
that would be a fantastic, like, motivating picture, right? Like before, one slide before. If we can further expand for the same event, we could have different, like, assay or platform dependent way to describe the same biological event. I'm wondering, like, for this fusion, would you have cytogenetic microscopic picture to show people?
Oh,
or
yeah,
no?
we have, yeah, we can find those in the literature, or we have those on hand in the lab too.
Yeah, what I'm trying to say is it's the same, like, fundamental biology event, but depends on the technique profiling platform you use, you get different like nomenclature.
Yeah,
Mhm.
absolutely. Plus, not only that, like this is just for your assays, right? Like not even talking about the knowledge that you'd get from a knowledge base, right? And how that could be represented.
Correct. Yeah. And so
I think this
is an
is,
Eve.
go ahead.
Oh, go ahead.
I was just going to say this is a great example because it's such a well-known event. So like BCR-able fusion is super common, but can be represented in all these different ways that might not be obvious to a clinician.
Right. And you would hope that somewhere in a patient report you would go back to just say it's
Yeah.
a BCR-able fusion.
Right.
But yeah, since all these different ways that this can be described. Okay. All right, and I was going to say also GA4GH VERSE group. So I know, Alex, you've presented this slide before. There are a lot of different, the reasons why we have a lot of different ways to explain these things are because we have different kinds of biomarkers, different kinds of assay technologies, and different kinds of variation calling methodologies. And so when we talk about an observed variation, we may have any reasonable combination here of a biomarker, an assay, and how we're calling that. And then being able to describe that may incorporate different ontologies depending on what methods are being used. Also, nomenclature, especially in cytogenetics, sometimes can be ambiguous. So if we don't know what it is we're seeing or what the genomic context or content is that are in a chromosome or some kind of genomic material, we might call it something like a marker or double minutes, which are like, they look like two little dots in your under the microscope. You may have a ring chromosome that's unidentified or a homogeneous staining region, which usually signifies an amplification, which would fall into our complex variants. Additionally, we might have an add chromosome. That just means we have additional material and it's at this location. We don't know what it is. Or we might not know what the breakpoints are, but we might say, this looks like a 16 and this looks like an 18, but I can't for sure tell you what the breakpoints are. And so there's a lot of ambiguity there too that is really difficult to understand and can it translate to any other ontology? Also, variant nomenclature is not always used consistently. I've included a couple different papers here just specifically on HGVS and the challenges in using HGVS for different kinds of variants and in databases. And then also, as I kind of mentioned before, variant nomenclature that represents a full characterization of a complex event, especially one that might contain multiple copy number variants or multiple junctions in a structural variant, often isn't super readable. So if I just gave you this nomenclature, it's a little daunting and it's not superhuman readable. So that can be a challenge. And then Cheryl has been helping kind of organize the knowledge-based challenges for the manuscript as well. And so some of the challenges in kind of gathering data for a knowledge base can be variant matching across different studies. Like I said, the same kind of issues across technologies, across genome builds. Some studies might use HG19, others might use HG38, some might use T to T. And then also a gene level matching versus the coordinate overlap. So we're looking at the two different ontologies, usually based on whatever assay type is being used to identify these. And then ongoing efforts to alleviate this have been things like the gene matcher and variant matcher that have been developed at Baylor. These can be used to look at your own variants across different ontologies and help to match them. And then also categorical variation is being, data models are being built by GA4GH. And so we could talk a little bit more about that too. And so a little bit about CatVerse. I know Wes is here, you could probably speak to this as well, but CatVerse really talks about categorical variation or aggregates of variants that fit a common description. So like a TP53 mutation or a TP53 deletion or a BCR-ABL fusion, for instance. And then the informational model can use different descriptive constraints and mapping to different ontologies to match these variants. So like MIC and gain could be matched with something that describes 5 copies of MIC-N, for instance. And so this is just a quick schematic that says there are different ways that information can be input and then matched based on these different constraints and mapping. So I don't know, Wes, do you have anything to add to that?
I think you did a good job with that. But yeah, a lot of it is then how to match and represent that data so then you can actually do different levels of matching between your search operations to make that actually feasible, which is nice.
Mm-hmm.
I
Yeah,
might add
a lot
one
of
more
work.
thing,
Yes.
which is CatVerse has been super helpful for representing some of the like common concepts we see across knowledge bases, like specifically canonical alleles was one of our major foundational use cases when developing this, which is used by like the ClinGen allele registry and the NCBI like SPEEDY format and their corresponding services like ClinVar. So having a way of representing that concept in a standardized way was really needed. There's no other standard that did it. So CatVerse helped us kind of close that gap.
Nice. And by canonical allele, what is meant by that? Can you expand?
Oh, oh yes, I'd love to. So canonical alleles are They're the notion of not just a specific variant on a genomic assembly, but also all of the associated context in lifted over assemblies, as well as projected out to transcripts that overlap that genomic region. And that set of variants collectively, like defined by the property of that genomic location and those operations, liftover and transcription, is the canonical allele. And there are services that will help you identify the canonical allele that are run by NCBI as well as by ClinGen that help you kind of link all those contexts to make it easier for you to do like duration and lookup and stuff like that.
Mhm. Great, thanks.
Yeah.
Was the canonical definition will evolve through the time?
No, that's a great question, and I think that's one of the challenges of the choice of term canonical. But that it is that's where the field is. It's what they call it. But one of the things we did with catverse is we actually define that originating context, right? So you basically, as the canonical allele provider, say what you mean as the founding canonical context on top of which you apply these operations to get to the set of variants. So it's really based off of what your definition as a knowledge base or service provider provide as canonical that defines it, and there's no one universal canonical.
Yeah, we have like thinking reference alternatively and minor and majorly, right? What things are conditional? Okay.
Mm-hmm. But I was just thinking in terms of like a complex variant, a canonical allele doesn't exactly apply, but catverse is super helpful in saying, okay, like MIC-N, I'm looking for a MIC-N gain or something. And then you have different types of information that may or may not align with that and help you match your variant. So it's helpful to understand like how it originated with the canonical allele in order to like help extrapolate that to what it might look like for complex variants. So cool, thank you. And then I just added these quick cartoons. Maybe you're able to read it in the last minute or so, but there's the challenge in matching different types of information that are seen through different assays and through different means or shared through different means. And then with verse and catverse, you can kind of match those a little bit better through a similar ontology or mapping method or constraints. And so I think that these are really great illustrations of what these products can do. Plus you get to see the little cat in the astronaut suit, which is awesome. And so when we're talking about knowledge-based infrastructure, some of the kind of roadblocks for advancement in this is that a lot of the work being done to populate these knowledge bases is very manual. And so even in commercial tools, as well as publicly available tools, curation of the literature, matching to results is still very manual and requires an expert with an understanding of interoperability between those technologies. And then also connecting complex variants and co-occurring variants can also be very manual. And this was something that we had touched on a couple of times in our working group as well. And then we can share our search knowledge using an interface or an API. There's different ways to do this. But if we use some of these tools that are being developed now, hopefully this will be more streamlined in the near future. And bottom line is there is a need to better communicate complex variants. So I don't know, Cheryl, do you have anything else to add on the knowledge base portion?
No, I think you summarized it well. I think there's the additional part of how the clinical information gets communicated, but and a large part of that is proper identification and proper matching to the literature based on determining what these alterations actually are and how they're commonly referred to. So
Mm-hmm.
we
And so
discussed
kind
that
of
as
the
well.
matching to the information can
Right.
be, yeah. Mm-hmm.
Matching to each other and then matching to the information and then communication of that information. Yeah.
Got it. Well, thank you.
Mhm.
So
Yeah,
currently
sure.
we're in the mid-
I still want to, use this opportunity to ask you. So for the information, as you mentioned quickly, there'll be the mention of complex variant and also the clinical, I don't know, the clinical phenotype, the disease. I mean, do you have different aspect of the information before you mention the information communication?
I'm not sure I follow your question, Chen. I'm
Yeah, what I'm
sorry.
trying to say is, okay, imagine one paper describe in a given clinical case or family or disease cohort, they find a certain complex variant. Then probably you will have at least two aspects of the information, which is genomic, right, description of how these complex variants were found. Then the other will be the clinical disease and the phenotype description.
Right.
So I was asking probably too quickly was, are there different aspects of your information need to be appropriately standardized before they can be put into the knowledge database?
Right. I mean, I think that largely goes to nomenclature, that you need to be able to know, there can be, it's not a lot of these variants and a lot of these structural changes um, will cause a similar phenotype even if they have different breakpoints. And so they can be, you want to be very specific about how you're identifying what that structural variant is, but you also want to be able to categorize it, catverse, et cetera, so that you can, because there could be a large number or, a lot of different fusions or structural variants that contribute to a single phenotype that's pretty uniform. Or the differences among those structural variants could lead to different phenotypes depending on which part of the genes they retain and which of the genes they retain if it's a larger chromosomal aberration. So it's very complex.
right. And I keep
I
thinking
still want
about
to add, like,
oh, go
for
ahead.
example, comparably to the genomic nomenclature, you want to have some ontology to guide your representation. Then to the disease phenotype side, do you want to use like phenotype ontology and disease ontology to guide the mapping or relationship, to the complex variant?
Yeah, I think it also depends whether you're talking about hereditary or constitutional changes versus somatic changes because the um inherited or constitutional change, really inherited changes, um, tend to be more uniform. They tend to be more founder type events, whereas the somatic changes can be much more disparate. So you can have um, different, structural variants that differ in their breakpoints and specific changes that are associated with the same disease ontology, but lead to different phenotypes, and that also depends on different um, other mutations that are present in that cancer. So it's I would say yes, we do need to have clear ontologies that we're using, and I think a lot of groups are converging on Oncotree and other disease ontologies, but um, I think there's not a one-to-one with
Hmm.
the structural variants,
And
if
really,
that
the scope
makes
of
sense.
our manuscript is looking at like the ontology describing the variants themselves. But
Right.
I think with the knowledge base, you almost have like three different ontologies. You have like your um, disease ontology, your oncotree, your context. You have the variant ontology that we're talking about now, and then you also have like your clinical significance kind of ontology. So I think of like, is the information we're drawing from this like related to therapy, diagnosis, prognosis, that kind of thing too. So it's
Yeah.
complex.
And I think about like heme diseases, for example, because I think that's maybe the best example right now is that a lot as like year upon year, the WHO and other classifying organizations, those like AML is not just AML, right? Now you have AML
Right.
with FLT3 ITD, AML with RUNX1, Run 1T1 trans fusion. And that RUNX1T1 fusion, again, can have, you need to make sure that you're rendering that so that you can make that diagnosis that this is an AML with RUNX1T1 fusion. It can be different depending on exactly how the pieces of DNA come together, but it is categorized as such. And then the disease mapping is to that very specific um,
Branch. Yeah.
branch. So, and those grow almost, annually. So,
Right. They can grow with like a couple papers, even
yeah.
especially in like things like sarcoma, where there are so many rare things that are showing up. There's so many different like very rare branches on those trees that, yeah, I feel like it's always evolving or will always evolve. Um, so yeah, that's cool. I like that. I like that thought. Um,
Yeah, thank you.
great.
I mean, that's very inspiring, but my thought and challenge is okay, then it's almost like you were shooting for the moving target, right? The disease definition and subtyping keep evolving. Then how would you at least say something? Maybe that's the common challenge you would need. How would you make the framework of knowledge base not like a keep after like being overwhelmed by, keep evolving definition rather than make it a flexible, expandable like framework. Just plug in whenever the new definition coming out and accepted by the guideline or the field.
Yeah, I mean, I think that's where you have to get into the categorizing and grouping. What is it? I think people talk about lumping and splitting, and I think you want to do a fair amount of lumping in order to be able to make these statements about what, you want to be able to give someone an understanding of like this, you know, unusual fusion is associated with this type of sarcoma, and it doesn't need to be this specific type of that specific type of sarcoma. You really need to make a decision about um how to describe it.
Hmm. Yeah, the whole lumping and splitting discussion, I feel, is more complex than meets the eye. All of this
Yes.
is more complex than meets the eye. It's all nuanced.
Yeah.
But yeah, that's all part of the challenge. So cool. All right. All right. So that's what I have for current work. For future work, the group had decided on focusing on Delins and hoping to come up with some um, with Delins and multiple co-occurring variants in cysts. We recognize that HGVS doesn't always work for communicating biological relevance. And so we were hoping to test a number of different instances of this and to help create a set of guidelines to help annotate these consistently. Because bioinformatics scholars may call it one thing which may not fully communicate biological relevance. And so we will look at this from all of our different standpoints that our group is so good at looking at a problem with, and we'll go from there. So here's one example of two KIT exon 11 deletions called by one of our NGS pipelines. So one is 8 base pairs in length, and the other is 13 base pairs in length with 4 base pairs of the expected sequence in between. And so the NGS pipeline called this as 2 frame shift variants in KIT. But when we combine this, it's actually a pathogenic in-frame Delins, which completely changes the meaning of what this variant would be. So the HDVS rules say to combine calls only if they affect the same codon, and that's not the case here, but I will tell you that our group did, because this was seen in a GIST tumor. So we wanted to make sure that this patient got the care they needed, and this is actually an in-frame pathogenic variant in exon 11. So we actually combined the call in our report and called it as an in-frame Delin. So that's all I have right now. But if you have any questions afterwards, we're happy to discuss. We still have another 10 minutes. If you want to shoot me or Chen an email, you can do that as well. But open to any questions, feedback, or discussion. So, and I appreciate that we've had a bit of discussion during the presentation too, which I found really helpful and enlightening. So, are there any questions?
Yeah. If no more question, I think sure. Still, I feel the most rewarding part or inspiring part was the discussion or the question answer with you. I feel your knowledge-based side, you may benefit having a overarching figure, just to indicate even the nomenclature and the complex disease complex and the evolving subtyping. I understand, the splitting or grouping by itself, it's a separate challenge. But if you can refer to that, the necessity, how we make the nomenclature interoperable, right? And then you can divide and conquer your splitting and grouping, disease specification. That'll be very inspiring because for each part, nomenclature bioinformatics and knowledge base probably we are over the words limit by 4,000 words in total as article. As and I checked last week for the JMIA requirements. So we were thinking and discussing having highly condensed and informative figure will be very beneficial for each part, speaking for their challenge and motivation rather than, go way over about the words limit. Anyway, I just want to say that's a very inspiring part to hear your explanation and why the knowledge base itself is so entangling with every aspect.
Thanks. I'll think
That's
about
why
that.
we need it. Yeah.
Yeah, this including Beth like triangle, Last week when Beth and I discussed about that, I was like, that triangle is too philosophical. How can we make it more materialize?
It's philosophic, but I feel like it launches our discussions in the right way. Like this
Yeah.
is inherently the problem. It's not an example necessarily, but it's yeah, it shows that as things get more complex, that disparate the disparence between like the clinical ontology and what might be a full characterization or something that we use on the back end is going to be quite a bit different.
Yeah.
Yeah. All right. Well, thank you everybody for joining. And we'll keep everybody in the loop with the progress in the manuscript. It's coming along. So we're hoping to have that submitted by the end of this year. So yeah. All right.
Yeah, really? Okay. Good luck to all of us.
Yeah. All right. Well, thank you all. Have a good day.
Thank you, you too.
Thanks, everyone.